Okay, thanks everyone. So welcome to the last session for today. And first up, we have Madan.
Yeah. Okay, thanks, Jitu. so, yeah, so that's right. So I think there's a change in title, and because I saw that I was inserted in the membranes section, I changed the title to this. Okay. I'm Madan Rao, and I'm a theoretical physicist at NCBS. And these are the two brilliant characters who did all the work that I'm going to talk to you about. What we've been working on for many, many years is how active stresses and active fluxes go to change the shapes and the molecular organization of all membranes in the cell. Okay, this is the plasma membrane on the outside of the on the boundary of the cell, which is subject to active stresses and fluxes from actomyosin cortex and lipid pumps called flippases. And the action of these gives rise to lateral and transverse organization at the plasma membrane. And this is something that I talked about in the last two lectures just a few days back. So today I'll be talking about internal membranes, and you see a picture of it here. Internal membranes such as the Golgi apparatus, endosomes, peroxisomes, etc., etc. These are membrane-bound organelles, and they're driven out of equilibrium by constant fusion and fission of tiny, tiny vesicles. Okay, so that's what makes it. These are instigated by GTP hydrolysis, and so they're active processes. Okay, and the questions that we ask. I got a whole slide on what are the various questions that we ask. Suffice at this stage to say that these internal membrane-bound organelles are subject to active fission and fusion. Okay. And that's what drives these membranes to show a lot of dynamics, both in their shapes, their size, and so on. And these are three pictures of the Golgi in different organisms. And you can see that there's a variety of shapes and sizes and numbers that they display. But the commonality here is that they're all subject to fission and fusion events. So as I said here, the Golgi is actually a system of cellular cisternae. Here you see 3 cisternae, 1, 2, 3, and they lie between the endoplasmic reticulum or the nucleus and the plasma membrane. Okay? And these are subject to fission and a vectorial transport of tiny, tiny vesicles, which fuse and fission out from these cisternae. And the system of cisternae exists in a constant flux of tiny vesicles that go back and forth. And this constitutes the steady state. And what we're interested in is what is the steady state size, the shape, the number of cisternae, the composition, and so on, in the presence of this finite flux. Okay, so that's the question that we're asking. There are many, many important cell biological questions that interest us, and they are listed here. Today, I'll be talking about what controls the size of the Golgi cisternae, what controls the shape, and I'll show that both the shape and the size, they depend on the state of the cell, okay, and they respond very sensitively to specific perturbations that might drive changes away from its homeostatic state. Okay, so here again is a schematic of the object of interest. The system of Golgi cisternae, here 4 of them, between the endoplasmic reticulum and the plasma membrane. Okay, and this is subject to fission and fusion of tiny vesicles that come out from here and move towards here. Okay, that's a live cell imaging of this entire process. Now, what you need to recognize is that fusion and fission involve a non-equilibrium mechanochemical cycle, schematized here as a 4-state Markov process. And these are driven out of equilibrium. So there's a current that goes through this Markov cycle. And the current that goes through the fusion and the fission cycle results in transport of membrane area and bulk volume. So the fusion will add a membrane area. The fission will remove membrane area. the elastic energies that need to be surmounted in order to fuse or fission tiny vesicles is around 70 kT. So you do require enzymes, active enzymes, to traverse this energy barrier. Okay, so this is the formulation of the problem. Oh, hi. This is the formulation of the problem. Again, a system of cisternae. Okay. So this is the model. Tiny vesicles come out from there's an influx of tiny vesicles at a given rate v, which come out from the ER, and then they fuse, they nucleate and grow, forming a cisterna of size M1. There's also fission from this cisterna, fusion again to form M2, M3, and so on and so forth. Okay, and what we want to write down is the dynamics of the probability distribution of having a cisterna, the first cisterna of size M1, second cisterna of size M2, third of M3, et cetera, as a function of time. Okay? The process, the dynamics that makes these the cisterna change size is fission decreases the size, fusion increases the size, and the increase and decrease of size is by 1 unit, given by the size of the tiny vesicle. Okay? It's known from experiments that the influx rate from the ER is quite fast. It could be between 1/10 of a second to 1 second. So this is actually a system which is driven out of equilibrium, because these timescales are much faster than the density relaxation time or the shape relaxation time of these objects, okay, of the cisterna. Because of the fusion and fission are fast variables, the updates of the sizes is much slower than the time scale over which the internal mechanochemical cycle happens. Okay? So there's a separation of time scale. Okay. So as I said, the question now is, how do we achieve stable structures of size M1, M2, et cetera, at steady state? Yeah? Okay. Now, I just wanted to make this very simple point clear. So imagine that you have an equation like so, right? That the time derivative of the size is some increases because of fusion, decreases because of fission. Okay? Then it's clear that the fusion and fission kernels should depend on size, because if they're not, and imagine that the fusion kernel is larger than the fission kernel, then this will give rise to an ever-growing size, yeah, with a fixed point at infinity. And if the fission kernel is larger than the fusion kernel, then it'll simply go to 0. Okay? So you require a topology of the kernels as a function of size to have a certain topology like this. For size where the steady state size is given by the intersection of the fission and the fusion kernel. For sizes larger than the steady state, fission dominates over fusion, and so it will shrink. For sizes smaller than the steady state, fusion dominates over fission, and therefore it will grow. Yeah? So this should be the topology, and indeed what we find from a more microscopic analysis is the following, that the fission and fusion kernels actually increase and then they saturate. With the fission kernel, you can see that the topology in the vicinity of this fixed point is like that. Okay. Okay. Yeah. So here is the equation for a single cisterna. This is the probability distribution of having a single cisterna of size M. In internal fusion cycle, so I and K label are the index for the internal states of the fusion and fission cycle respectively. Okay. So in this particular calculation we have done, I goes from 1 to 4 because it's a 4-state Markov cycle. And this is a function of time. Okay. So this is just a master equation. There are gain terms and loss terms. And only after you go through one complete cycle do you add or subtract mass, depending on whether there's a fusion or fission. Okay. Now, assuming that the internal state dynamics is fast, you can use something called an adiabatic approximation, and you can get an equation for of this simplified form, where these K fuse and K fis represent the mass-dependent kernels of fusion and fission, respectively. Okay. Now it turns out that if we sort of, was this the cycle? Yeah. If we sort of take these internal states seriously and put in all the physics and the mechanochemistry behind each of these internal states, then this turns out to have, these kernels turn out to have a very large parameter space. Okay, that is, you need to have some 30 parameters that describe it. Okay, and that's crazy if you're ultimately describing a curve like this. Okay, so there's clearly a dimensional reduction that one can do, and we have done that in a sort of systematic way. And one arrives at a smaller set of parameters. I'll show you 3 parameters. Okay, that describes the mean field dynamics of how the mass changes as a function of time. Okay, how the first moment of this equation changes in time. Okay, so that's the equation. This is the equation for how the size of the single cisterna, the size of the single cisterna changes with time. Nu here is the influx rate from the endoplasmic reticulum. A0 is the nucleation rate, which describes de novo nucleation and growth of cisterna. This full thing here is the fusion kernel. Okay, and C1, the parameter C1 tells you what is the limiting levels of fusion enzymes. Remember, these are enzymatic processes and require a certain enzyme availability. Okay, so this is the C1 represents the total amount of fusion enzymes that you have in the cell. This here is the kernel which describes fission, and C2 here denotes the levels of fission enzymes. Okay? The parameter D2 is the effective fission rate. Okay? So that's the equation, and you can see that there are much fewer parameters than what we started out with. Okay. Now the entire physics, of course, the fixed point. So this is now a dynamical system, right? And we can analyze this as a dynamical system. The fixed points of this dynamical system depend on the intersections between the fusion and the fission kernel, and they are depicted here. So you can see that you can either have one stable fixed point, or 2 stable fixed points, or no stable fixed point. Okay, and that gives rise to the different phases that you see here. So what I'm plotting here are the levels of the fusion enzymes versus the influx rate. This shows a phase diagram where the influx rate is very tiny and the fusion levels, the levels of the fusion enzymes is low. Then you are in a phase where you don't have any large structures, they're tiny vesicles. When you go beyond a certain threshold, you start getting stable cisterna of mass whose size increases with the influx rate. Okay? When you have very large influx rate, then of course you get unbounded growth. Because there's no stable fixed point that you see here. Okay, then, now it turns out, I mean, you can see easily that for these values of the Hill coefficients, alpha equals 2, beta equals 1, you can rewrite this as this dynamics as a purely relaxational dynamics with a free energy which has this form. Okay, so if I'm starting with I'm starting with a de novo, that is m starting with 0, then you see that little bump there means that there's a very slow initial decay, decay into this phase. Okay, so it'll start growing extremely slowly, yeah, because of the presence of that little maxima there. Yeah, I think this is all I wanted to say about this. The cisternal size that which is the size at this stable fixed point, is dictated primarily by the levels of the fusion and fission enzymes, and it has a certain dependence that can be arrived at analytically. Okay? So this is one of the predictions of the theory about how the cisternal size depends on the levels of fusion enzymes and fission enzymes. Okay. So now let's go to what happens when you have 2 cisternae. Okay. So this is the picture that you have, the endoplasmic reticulum. Tiny vesicles emanate from the endoplasmic reticulum, fuse to form the first cisterna, fission from the first cisterna, form the second cisterna. The second cisterna can give tiny vesicles to the first one and also get depleted by transferring tiny vesicles into the plasma membrane. Okay? And you write down similar equations that I wrote down earlier for the sizes of the cisterna 1 and cisterna 2. subject to the dynamics that is schematized here. Okay. Now, there's a simple general statement that one can make here, and that's the following. Imagine that you have the dynamics like so, okay? So this is the time dependence of the first cisterna, time dependence of the size of the second cisterna. That is fusion terms, fission terms, and this is a term which comes from intercisternal transport, 2 going to 1, okay? Similarly, M2 has fluxes coming from cisterna 1, this arrow, loss coming from, because it goes that way, and there's further loss here coming from the transfer to plasma membrane. Okay? So with this kind of dynamics, one can show the following, that if these intercisternal kernels are only dependent on the donor membrane, then this system of dynamical equations can only give rise to fixed points and no limit cycles. Okay? In order for you to get limit cycles, you require that the intercisternal transport, namely 1 going to 2 and 2 going to 1, should depend both on the donor membrane and on the target membrane. Okay? That's an important point, and I'll come to the significance of the statement later. Now, how do you achieve that? And you achieve that because the intercisternal vesicles that go from 2 to 1 or 1 to 2 have to carry what's called V snares from the donor cisterna and T snares into the target cisterna, and only then will appropriate fusion happen in the target. Okay? Yeah, so that's the molecular basis for that statement that I made, that H1 and H2, these two terms, have to be functions of M1 and M2 in order to get limit cycles. Okay? Okay, so here is an example of H1 and H2. So you have 2 cisternae betwixt the endoplasmic reticulum and the plasma membrane. You have transport from the ER, intercisterna transport, and transport into the plasma membrane from the second cisterna. The dynamics of M1 and M2 is specifically given by these terms. Here you can see is the intercisternal, going from 1 to 2. This is from 2 to 1. Okay, so we can now ask, what is the phase diagram that comes out from analyzing this system of equations? Okay, and it's a more complicated phase diagram. This is the influx rate, and D2 is the intercisternal rate. And you see that apart from the fact of having vesicles and stable cisterna, that is having 2 cisterna that is stable, you also have limit cycles. Okay, so in this region of parameter space, you have stable limit cycles that are a consequence of a Hopf bifurcation. Only 5 minutes more. Oh, wow. Okay, okay, and that's the picture shown here. But I want you to look at the look at these oscillatory phases. The oscillations of the sizes M1 and M2 can either be in phase, like shown here, or out of phase, like shown here. And in fact, they can be in between these two also, depending on the choice of parameters. Yeah? You can calculate so now this one, I want to identify with a well-known, or maybe I'll come to that later. Okay, so now, okay, so what happens here? This is showing that there's a periodic dissolution of the cisterna. The cisterna grow together, M1 and M2 grow together, and they fragment together, grow again, and so on. So if I entrain this with the cell cycle, then this periodic dissolution of the sizes can happen during mitosis. At every cell cycle event, it can happen. Yeah, that's one, and that's what I'm going to do here. But I don't have the time to get into the details here. This here is a very interesting phase. This says the first cisterna, let's say, grows, then it decreases, then the second cisterna grow, that decreases, and so on and so forth. Okay, so it's an out of phase thing. Now this is, for those of you who know the controversies that have gone on in this field, this is related to what's called a cisternal progression. Okay. Okay. So there are 2 contesting models that people look at. One is called vesicle transport and cisternal progression, and they are taken to be opposing models. Okay. However, what we are saying here is that the in-phase solution corresponds to this, the vesicle transport model. The out-of-phase solution corresponds to cisterna transport. So they're not two different models. They're the same underlying physics, but they're two different phases of the same underlying. Yeah. Okay. So now, as I said, if you entrain this with the cell cycle, and so you write down an equation like so, where the influx coming from the ER has a periodic signal, then you find that the period of the dissolution of the cisterna gets locked in with the cell cycle. Okay, and so this is again one of the predictions that comes out from this. Okay, but I think the interesting part here is that this simple dynamical system and dynamical analysis shows that the form of the Golgi compartments actually depends on the cell state. Okay, and that's the interesting thing, because it depends, the cell state tunes the value of the various parameters, including the influx coming from the ER. They're governed by kinases, which are coupled to them. Okay. I think how much time do I have now? Right. Okay. 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 So using this, this theory allows us to do following. So far, I've been talking about mean field equations that arise. Mean field equations that allows us to do a dynamical systems analysis. You can ask what is the effect of noise, and the noise could be either extrinsic, that is there in the influx rate, or that it could be intrinsic, that is there in the chemistry that defines the stochastic Markov process. Yeah. And you can ask what happens to the system of equations when you introduce these various kinds of noise. And you can what we can do is to analyze what is the regime of stability of this dynamical system to these kind of noise. The other question that you can ask is what happens if I make very specific predictions. What happens if I cut off the supply of influx from the ER? What happens to the cisterna? What happens if I fragment the last cisterna, the trans cisterna? What happens to the whole structure? And it makes very specific predictions about the time dependence of the sizes of these structures, which should be tested experimentally. So I'm hoping to, so in fact, I was talking to Linda and saying that I'm really looking forward to talking to an experimentalist who does live cell imaging on the Golgi to test some of these ideas. So if any of you know such a person, then please point them to me. Okay, now I was going to talk about shape control, but I think I don't have that time, except to say the following, that just look at this picture. We talked about this before, the two models, namely the vesicle transport model shown here and the cisternal progression model shown here, thought to be previously two different models, are from a dynamical analysis, different solutions of the same underlying physics. But once you see this kind of picture on the right, then the obvious question that one should ask is, what is the driving force for the movement of the cisterna? Okay? And that's when, and that's the subject of this part of the talk, which I won't get into. But the result of our analysis is to actually provide an answer for what is the driving force. And the driving force is indeed the non-equilibrium forces that arise from fission and fusion. And I'm just going to not go further in that. And I'll just end the talk. So those are the set of people who did the work and the funding agencies and the various papers that have come out.
Yeah. So you said at the end that you want to test predictions with the by perturbing some things. So is it, when you do that, it is possible that those experiments would be affected by some of those parameters that you remove? I mean, you reduce the dimension.
That's right.
So maybe some parameter in those parameters might get affected in these experiments.
Yes.
So how would that be predicted from here? Because would that cover the prediction?
No, so I mean, the input will be these parameters, and they are molecular parameters. They have to do with the levels of the enzymes, which we will know.
Okay. They will effectively affect some of these parameters.
Yeah. So they'll make you move around in this parameter space. Okay. Yeah. But at least, qualitative pictures can be obtained, right? I mean, for instance, one can ask what happens if you shut off the influx from the ER. That's an easy experiment to do. And then you measure the mass of or the size of the Golgi.
I was thinking, what is the cost that you pay by reducing the dimension? Because is the experiment happening in this whatever the full parameter space?
Yeah, but there's always parameter redundancy. No? So the fact is that you have a curve which looks like that can be fit by 2 or 3 parameters. So there's a lot of combination of parameters which might give you the same.
Let's go on. So Linda.
So Madan, the levels of, do you know if the levels of the fusion and fission enzymes actually do vary?
In different cell types?
Yeah, or even like during the cell cycle?
Right.
They're
So
influenced by.
Right. So I think what I've read is that these the levels, the influx rate is governed by a variety of kinases. Many of the, for instance, the influx rate from the ER, the fusion rates into the Golgi, and many kinases are under the control of cell cycle proteins. This is all I know. I mean, it would be good to really get a quantitative understanding of these parameters because they are crucial.
Because they're so important to your equations.
Yeah.
Then the other question is there a typical number of cisternae?
Yeah.
It's typically 4 or what?
Yeah. So that was the set of things I didn't talk about. Yeah. That's a question that I'm very, very interested in. So that's the variation. There are around 4 or 5 in human cells. In plants, around 20. In green algae, for some reason, there are around 100. So the question now, of course, is what controls the number of the cisterna? And we have studied this problem, and our take on it is that is actually governed by the function that the Golgi is supposed to do. The Golgi is supposed to make post-translational modifications to a lot of proteins. It sort of adds sugar groups on proteins and then displays it on the cell surface. Those are called glycans. And the number of cisterna is related to the complexity of the glycan code. That is. Yeah, because in green algae, the glycans do a variety of things. They're associated with the immune system, cell attachment, and various things like that. In plants, they do many more things than.
Madan, what decides the shape? Is it just the surface to volume ratio because you know how much fusion and fission is happening, or is it also dynamics?
It's also dynamic. Yeah, it's so the way we analyze it is to ask what is how does the membrane area change as a function of fission and fusion, so that when you under fusion the area increases and the fission the area decreases. It will also change the volume. So that those are the 2 quantities but it also applies forces. So there's a force dipole associated with each fission and fusion event. And so it's a combination of these 3 things that gives rise to a shape.
I just have a question. I just have one quick question, which is related to the first. So you mentioned that the switching between the 2 states could be governed by the cell cycle. So are you able to sort of make specific predictions? For example, if you delayed cell cycle through drug treatment, which is quite straightforward.
Yeah.
Can you make specific predictions in the model for what you think would happen?
We could. We haven't done that yet, but we could. Yeah. I mean, but the thing is that I think it's related to Linda's question about what are the various things that are affected by cell cycles, right? This I don't know. So there are many parameters in the theory. In the effective models that we have, there are a few parameters. But even so, the only what we have taken is that the influx rate is governed by the cell cycle and nothing else, but that may not be correct. I think,
yeah, okay. So then, are you going? Yeah, okay.
I'm just confused. I mean, so the pictures you have here are for what?
This one, that's a plant.
The one on the right. I mean,
the one, these 2 are plants.
I see. They are plants, right?
Yeah.
But I mean, I see. So I mean, the thing is that when you have so many of them, it looks like it actually wants to form a periodic layered phase. Whereas if you have only 3 or 4, it just looks like it.
Yeah.
So is there some tendency? I mean, should one think of it in some limit as a whole stack, or is that just my fevered imagination? Is there
I
a
don't know,
period,
but
and is
just
there
saying that you can think of it like a crystal and
a laminar phase?
Okay.
I mean,
I
at
don't
least on the
think so.
right.
Huh?
At least on the right, it sort of is heading that way. So
Yeah.
is there a good reason to think of it that way? Or is it just my
So, okay, in some sense, a lot of people think, a lot of biologists think that everything is governed by proteins that reside on this, right? So there's a stack of proteins that people call Golgi, whose periodicity defines the periodicity of this. I mean, I find that a bit, I find ascribing everything to very special purpose of proteins.
I'm only saying it's layered. I'm not saying that it's.
I don't know, but I'm just saying that thought. I doubt whether there is. So, for instance, if you get rid of a lot of the proteins that on the cytoskeleton, this sort of starts drifting away. Okay, the layers start drifting away.
For the sake of time, let's thank Madam. What's that?